A very good evening to all of you. Today we will be doing biochemistry. So let's begin with the 16 questions are fixed in Neet PG for biochemistry, and in INICT at least 12 questions. So let's begin with the biochemistry. ApoB48 is a remnant of A. RNA editing, B. RNA interference, C. RNA splicing, D. DNA translocation. Well, the correct answer here is RNA editing. What happens in liver cells? ApoB100 gene gives rise to ApoB100 protein, but the ApoB48 protein is made in intestinal cells from the gene encoding ApoB100 by the process of RNA editing, which is also known as differential RNA processing or chemical modification of RNA. This is post-transcriptional modification. Now, this RNA splicing is also a post-transcriptional modification, but this is the removal of intron and not editing. So the correct answer here is A. Hepcidin decreases iron absorption by inhibiting A. Divalent metal transporter, B. Hephistin, C. Ferroportin, D. Transferrin. Now this hepcidin regulates the iron metabolism. It mainly regulates iron entry in circulation. By inhibiting ferroportin, hepcidin reduces dietary iron absorption. The iron release from macrophage is also reduced by ferroportin inhibition. So increased hepcidin activity is responsible for reduced iron availability. Increased hepcidin will lead to anemia. So the correct answer here is C, ferroportin. Which of the following has the highest glycemic index? A. Glucose, B. Fructose, C. Sucrose, D. Sorbitol. It is glucose. GLUT5 is a transporter for A. Galactose, B. Fructose, C. Mannose, D. Glucose. It is for fructose. Which of the following is insulin dependent? A. GLUT1, B. GLUT2, C. GLUT3, D. GLUT4. It is GLUT4. Only GLUT4 is insulin dependent. Insulin recruits GLUT4. GLUT4 is present on the membranes of peripheral tissues, for example, skeletal muscle, cardiac muscle, and adipose tissue. It helps in insulin dependent. Cause of HHH syndrome, that is hyperornithemia, hyperammonemia, homocystinuria, is due to A. Deficiency of ornithine transcarbamylase, B. Defect in ornithine transporter, C. Deficiency of ornithine aminotransferase, D. All of the above. It is defect in ornithine transporter. Defect in ornithine transporter leads to HHH syndrome, hyperornithinemia, hyperammonemia, homocystinuria. This prevents ornithine transport into mitochondria and hence in the cytoplasm, and its reduced ability to clear carbamyl phosphate and ammonia. Which method is used to separate a mixture of lipid? A. Isoelectric focusing, B. Page, C. Chromatography, D. Electrophoresis. It is chromatography. Wrinkled paper appearance of macrophage in bone marrow aspirate is seen in A. Tay-Sachs disease, B. Gaucher's disease, C. Niemann-Pick disease, D. leukodystrophy. It is Gaucher's disease. Wrinkled paper or crumpled tissue appearance of macrophage in bone marrow aspirate is seen in Gaucher's disease. Go to the description. A link is there. Just click the link and join the Ultimate Neet PG Breakthrough Blueprint Masterclass covering all the 19 subjects in a holistic manner in just 3 days. The first 10 people who click the link will get this masterclass absolutely free. In CRISP or Cas9 system, which repair mechanism is used for genome editing? A. Homologous repair, B. Non-homologous end repair, C. Mismatch repair, D. Nucleotide excision repair. It is non-homologous end repair. A rapport Lubick cycle shifts oxygen dissociation curve to right because of A. 2,3-DPG, 1,3-BPG, 3-phosphoglycerate, 1,6-phosphate. It is 2,3-DPG. Hot flashes are experienced as a result of increased noradrenaline, decreased estrogen, increased noradrenaline and decreased estrogen, it is increased noradrenaline and decreased estrogen. Restriction fragment length polymorphism is used for Analysis of DNA fragment, DNA stems, synthesis of nucleic acid, detecting proteins, analysis of DNA fragment. Thermogenic food in which of the following? High protein diet, high carbohydrate, high fat diet does not depend on macronutrients. High protein diet. Ketone bodies are not utilized by A. Brain, B. RBC, C. Heart, skeletons. So they are not used by RBC. In RBC, there is no mitochondria and ketone body utilization pathway takes place in the mitochondria. RBC cannot utilize ketone body. Brain, muscle, heart can use ketone bodies during digestion. Enzyme deficient in hyperammonemia type 1 is ornithine transcarbamylase, EB arginase, C carbamyl phosphate synthase, arginosuccinate. It is carbamyl phosphate synthase. Type of mutation seen in sickle cell anemia is A. Insertion, deletion, point mutation, frame shift mutation. It is point mutation. Which replacement of amino acid does not alter its normal function? Glutamine replaced by asparagine. The clinical feature given below occurs due to deficiency of tryptophan, phenylalanine, cysteine, glutamine. It is tryptophan. Which of the following binds mRNA with ribosome in eukaryotes? Poly-A tail, tRNA, 7-methylguanosine cap, shined aluminum sequence. It is 7-methylguanosine. Which of the following is anaplerotic reaction? Conversion of pyruvate to oxaloacetate. Which of the following, very important question, group of protein assist in folding of other proteins? A. Proteases, B. Proteasome, C. Template, D. Chaperone. It is chaperone. Myoglobin has which structure? Primary, secondary, tertiary. It is primary, secondary, tertiary. Best investigation for metabolic disorder is Western blot, tandem mass spectrum, PCR, electrophoresis is tandem mass spectrum. Separation of protein by their mass is by A. Gel filtration chromatography, ion exchange chromatography, isoelectric focusing, affinity chromatography. It is gel filtration chromatography.